This module will focus on gene expression and will explore basic concepts related to understanding the genetic code, as well as the processes of transcription and translation. Let's begin with an understanding of the so-called central dogma of molecular biology. We learned in a previous module that DNA is responsible for its own replication. So on one hand, DNA can replicate itself, but DNA is also responsible for other important parts within a living cell as well, especially information flow. So DNA, in addition to replicating itself, can also code for specific messenger RNAs. So here we see a small piece of messenger RNA, shown in yellow, which was coded for when a specific gene was expressed. That messenger RNA then leaves the nucleus of the cell. That RNA then interacts with ribosomes, and the bases in the RNA are translated into a specific protein. So ultimately, this specific sequence of amino acids in a protein is dictated by the sequence of bases in messenger RNA, which was originally dictated by the sequence of bases in DNA. So the process of converting the information in DNA to RNA, we refer to that as transcription. And then translation refers to translating the sequence of bases in messenger RNA into a specific protein. Let's explore the genetic code itself. It was back in the 1950s that Watson and Crick published the structure of DNA. Shortly after that, in the 1960s, research was done which essentially cracked the genetic code. The results of that research are summarized here in this table. So in essence, what researchers discovered was that the sequence of bases in messenger RNA in groups of 3, referred to as codons, those codons code for specific amino acids. And also, keep in mind, when we look at a genetic table like we see here, that codons are typically written in a 5' to 3' direction. So for example, how is this genetic code used? Imagine, if you look toward the bottom of the screen here, imagine a situation where we have a section of DNA, illustrated in orange, double-stranded molecule. So notice the anti-parallel nature of the DNA bases here. Let's take, for example, this bottom strand of DNA, for convenience illustrated in a 3' to 5' direction. Those would, if transcription occurs, those bases would produce a messenger RNA, shown in yellow, with the corresponding complementary sequence of bases we see here. And then the bases in that RNA are used to code for specific proteins, or specific amino acids. So for example, notice AUG, that's a group of 3 bases, that's a single codon. If you look at the table, AUG codes for methionine, that's a start codon. That represents the start of the formation of this specific protein. If we keep going to the next codon, GCC, that codes for alanine. AAU, asparagine, and so forth. Notice a few things to keep in mind, that the genetic code is redundant and specific. What does that mean? Well, it's redundant in the sense that these amino acids, phenylalanine, leucine, serine, proline, notice that the amino acids are coded for by multiple different codons. It's specific because every specific codon does indeed code for a specific amino acid. So what are the implications? Well, what this means is that mutations can occur in DNA which can potentially lead to changes in RNA bases, but that doesn't necessarily lead to changes in specific amino acids. It might, but it might not. Okay, these so-called silent mutations are ones that occur when there's a change in the DNA base itself, but there's no overall change in the amino acids that's produced at a specific region of a specific protein. So that's kind of an overview of the genetic code. Now let's take a closer look at gene expression. Let's focus initially on transcription. In other words, converting the information in DNA to a corresponding piece of RNA, specifically messenger RNA. We can identify 3 stages of transcription, namely initiation, elongation, and termination. But in essence, what is occurring during the process of transcription is this enzyme, RNA polymerase, is essentially identifying a specific region of DNA. It then makes its way down that portion of DNA, and the RNA polymerase brings in complementary RNA bases to form a piece of messenger RNA. So again, RNA polymerase, a key enzyme involved in the process of transcription. Imagine the DNA within your cells consisting of billions of base pairs. How does RNA polymerase know where to begin? Because after all, there are thousands and thousands of genes, but of course not all of these genes are expressed all the time, and of course they're not all expressed in all of these cell types in your body. So how does RNA polymerase know where to begin transcribing a gene? Well, if we look at the first phase of transcription, namely initiation, we'll first of all introduce this concept of a promoter. A promoter or a promoter region, that's simply a region of DNA that contains a specific sequence of bases where the initiation of transcription takes place. So essentially what happens at the promoter region is that there are these various other proteins, we generally refer to these as transcription factors, that bind to a promoter region. So in this image you see this little gray sigma here. You might say, well, that's a transcription factor. It's just a specific protein that binds to the promoter region. Once the transcription factors bind there, that then brings in RNA polymerase, causing RNA polymerase to bind to that region. That then begins the process of transcription. So transcription factors, again, play a key role in initiating the transcription of specific genes. So once transcription factors are in place, RNA polymerase is in place, then the elongation phase can occur. Essentially all that happens here is the RNA polymerase makes its way down, again in a 5' to 3' direction, makes its way down the DNA, producing a piece of messenger RNA as it makes its way down. That continues for a while, but at some point the RNA polymerase must know when to stop. After all, it doesn't make its way down the length of all of your chromosomes. It only transcribes a certain portion of that DNA corresponding to a single gene. So how does the RNA polymerase know when to stop? Well, it turns out there are specific sequences that when transcribed, if you notice the image toward the right here, notice the messenger RNA that's produced in yellow. When specific sequences are transcribed, we call these the terminator sequences, they produce these regions of messenger RNA that fold back upon themselves, and they form these little hair loop type structures. When those are formed, that triggers the messenger RNA to release itself from the RNA polymerase, shown in green, thereby ending the transcription of that particular gene. So now we have this piece of messenger RNA shown in yellow, but that's not quite the end of the story. It turns out that the RNA has to be processed a bit before it can be used in translation. So we have at this point what we call a primary messenger RNA transcript, or simply pre-messenger RNA. So now what happens? Well, again, the RNA is processed a bit. A few things I want to point out here. If you look at the RNA that's produced, and even the DNA itself that it was transcribed from, it turns out we can identify these regions referred to as exons and introns. Introns, it turns out, are regions of the, for example, the primary RNA transcript that are removed, so that the exons are left in place and they're essentially spliced together to produce a mature, well, I shouldn't say mature quite yet, but a further mature piece of messenger RNA shown toward the bottom of this image. So again, exons are portions that are left in. Introns are those portions of the RNA that are removed before the RNA is used for translation. In addition, there's a bit more processing that occurs. Let me first, however, mention the implications of this splicing of exons. Really quite interesting, because it turns out that a single gene, illustrated in region A here, a single gene can have multiple exons that can be spliced together in different fashions so that a single gene can produce several different types of proteins. So that's, again, it's kind of a big deal, right? A single gene can produce multiple messenger RNAs and therefore produce many different types of proteins. Okay, so how else is the primary messenger RNA modified before it's actually used for translation. I'll just quickly mention messenger RNAs tend to have these so-called 5' caps, 3' poly A tails added. These are simply regions that help target this messenger RNA for translation and also prevent the messenger RNA from being broken down immediately by specific enzymes. So after all this processing of this messenger RNA takes place, then what happens? Now we have a mature messenger RNA that's essentially ready to be used for translation. So the mature messenger RNA essentially now leaves the nucleus and is ready to be translated into a polypeptide, or essentially into a protein. So translation, this next major phase of the central dogma of molecular biology, let's talk briefly about some of the key players involved in translation. First of all, we have the messenger RNA that we just discussed that was formed from transcription. We also have some other types of RNA, specifically transfer RNA or tRNA, shown in the two images here. A few things about these tRNAs. Notice that a mature transfer RNA is bound to a specific amino acid. So toward the right, we see a transfer RNA connected to the top region here is the amino acid serine. Also notice that there are 3 bases located in a key position toward the bottom of these images. These 3 bases represent an anticodon. Anticodons are complementary to the codons found in messenger RNA. So in essence, what these tRNAs do is they bring specific amino acids into the system that are then linked together to produce fully mature proteins. So we mentioned mRNA, tRNA, and then finally the major structures involved in producing these proteins are the ribosomes. So as we'll see in just a moment, the ribosomes interact with the messenger RNA. They bring in the appropriate tRNAs, each carrying its specific amino acid, and therefore these are structures involved in the formation of proteins. So we can also identify 3 stages of translation. Let's first of all focus on initiation. It turns out that ribosomes, these are structures found within cells that contain very specific proteins. They also contain some ribosomal RNA, or rRNA. It turns out that ribosomes consist of 2 subunits. So if you look toward the right in this image here, you see this large unit colored orange, as well as a smaller subunit toward the bottom. So these small and large subunits of ribosome, they come together and they combine near the stop codon. So again, here's our messenger RNA. Somewhere near there's a start codon, and this is the region where the ribosomes bind to the messenger RNA. So they bind near the start codon. Once the small and large subunits are in place, then essentially we have the elongation phase of translation. So essentially what happens is the intact ribosome, shown in yellow, makes its way down the messenger RNA, again in a 5' to 3' direction. As it moves its way down, it reads the codons in the messenger RNA, bringing in the appropriate tRNAs, whose anticodons are complementary to the codons. Each tRNA then, of course, carries a specific amino acid. Bonds are produced or formed between these amino acids, and they're then released from the ribosome, ultimately to produce the fully formed polypeptide or protein. So, finally, how does the ribosome know when to stop translation? Well, the third and final stage of translation is termination. So recall when we discussed the genetic code, you might have noticed some stop codons. So essentially what happens, there are a few different stop codons that actually code for not for specific amino acids, but they code for something else. And essentially when the ribosome encounters these stop codons, that terminates further formation of this long chain of amino acids. So when the ribosome encounters a stop codon, that's the end of translation. Ultimately, that chain of amino acids is released, a fully formed protein eventually is produced, and essentially the product of gene expression, namely the protein, is complete at this point. So that concludes our brief coverage of gene expression in living cells.